So each myosin thick filament, it contains approximately 300 myosin protein molecules. ATP is produced in the globular head of the myosin protein. So muscle contains a red colored oxygen storing pigment called as myoglobin. When they are in the contraction state, the actins will, or the myosin will go and bind to the actin there. So that is when the contraction occurs. Hello everyone, a warm welcome to another session on chapter 20, that is locomotion and movement. I am Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. In the previous session, we studied about the structure of the skeletal muscles, the visceral or the smooth muscles and the cardiac muscles. In today's session, we shall study about the function or the structure of the contractile proteins. So, contractile proteins are the ones that help in the contraction of the skeletal muscle. So, we shall study about the function and we shall learn about the mechanism of the contraction of the muscles or movement of the muscles. So, to begin with, we shall start with learning about the structure of the contractile protein. So, in the previous session, we had studied about the anisotropic band or the A band, also called as the myosin filament, which is made up of the myosin protein, and also we learnt about the light band, which is also called as the I band or the isotropic band, and it is also called as the actin filament as it is made up of the actin protein. So, what exactly is the structure of the actin and the myosin protein we shall look into. So, each actin, so actin is a thin filament, right? So, it is a thin filament, it is light pink in color and it is also called as the isotropic band or the I band. So, each actin filament is made up of two F actins, which are called as two filamentous actins, which are helically wound to each other. So, if you see the image here, you can see they are made up of two filamentous actins. That is the F actin, or they are called as filamentous actin. And they are helically wound to each other. What do we mean by helical? Helical is something, but if two lines are there like this, they are parallel to each other. But if I draw the lines like this, wherein they are coiled to each other, it is called a helical structure. So, if you can see here, just like the DNA is also helical structure, right? Just like that here, the actin protein is made up of two F actin filaments or filamentous actins, which are helically wound to each other or coiled to each other, and each F actin is a polymer of monomeric globular actin. So, what are they made up of? Can you see they are a polymer, right? Polymer means they are like chains. So, how is the F actin? They are in the form of chains like this. So, when they are in the form of chain, they are called as polymer. Now, when I say monomer means I am considering only this. So, each polymer of F actin is made up of a monomer of globular actin. Globular actin. It's easy to remember. They are globe-like. They are like a ball, like a circle, right? So, that is why they are called as globular actin. So, each of these globular actins, they come together, they are joined together to form chains or polymers, which are called as the filamentous actin. And two filaments of another protein, which is called as tropomyosin, also is present close to the F actin throughout its length. If you can see here, these are the tropomyosin. So, they are present very close to the, they are also proteins which are present very close to the F actin. So, therefore, so far we understood that structure of the contractile protein. We were studying about the structure of the actin filament or the actin protein. So, what is the structure of the actin protein? Actin proteins are made up of monomers of globular proteins. These monomers of globular proteins are held together to form a long chain called as the filamentous actin, which are coiled in a helical manner, and apart from that, very close to the filamentous actin is present one more protein which is called as tropomyosin. So much we have understood about the structure. Next, apart from tropomyosin, there is a complex protein which is called as troponin, and it is not present everywhere. It is only distributed here and there at regular intervals on the tropomyosin. So if you can see the tropomyosin, regular intervals here and there on the tropomyosin, there is presence of the troponin. So troponin is also one of the protein that is present, and whenever the muscle is in a resting state, or whenever the actin filament or the isotropic band or the I band are in the resting state, a subunit of troponin masks the, or it completely covers the active binding sites for myosin on the actin filaments, and that is how they come back to the relaxation state. So when they are in a resting state or in the relaxation state, there is subunits of troponin, right? Where are the troponins present? They are present here and there, distributed on the tropomyosin protein. They actually cover or they mask the binding site of the myosin. Why? Because the binding of myosin is also important, right? Because if the binding of myosin will occur, then contraction will occur. That should not take place. So if relaxation has to occur, then the binding of the myosin should be avoided. So that binding of the myosin is avoided when the muscles are in the resting stage by a protein called as troponin, which is distributed here and there in regular intervals on the tropomyosin protein. So this is the structure of the actin filament or the actin protein. Next, moving further, we'll study about the myosin protein. Myosin filament, it is made up of myosin protein, and this filament is also called as the anisotropic band, or it is called as the A band, or it is also called as the thick filament because they are thicker when compared to the actin filaments, and these myosin filaments are darker in color. So each myosin thick filament, it contains approximately 300 myosin protein molecules. Again, these are polymers, but they have shown here a monomer. Just one myosin protein molecule they have shown here, but many myosin protein molecules are held together to form a polymer or chain-like structure. But how many monomers of myosin proteins are there? There are 300 monomers of myosin proteins molecules, and many monomeric proteins called meromyosin constitute one thick filament. So many monoproteins or many monomers of the myosin protein, they come together, they join together to form one thick filament of the muscle fiber, and it is called as the meromyosin. Now, one monomer is called as the myosin protein, and chains of myosin protein is called as meromyosin. So each meromyosin has two important parts. It has a globular head, and it has a short arm, and it has a tail. So if you can see here, the presence of the globular head, it is globe-like, and it has a tail. This part, it is the tail part, and it has a arm. So these are the structures or the parts of the meromyosin. And the former, that is the globular head, being called as the heavy meromyosin, and the tail being called as the light meromyosin. So the head part is called as the heavy meromyosin, and they are the actin binding sites. So actins, when they are in the contraction state, the actins will, or the myosin will go and bind to the actin there. So that is when the contraction occurs. So this is the actin binding sites, and also the globular head part of the meromyosin is the ATP binding sites for the muscles. So this is about the structure of the protein. And next, moving further, the heavy myromycin component, that is the head and the short arm. So we had studied, right, this head part along with the short arm, it is the HMM. It is the heavy myromycin component. Whereas the tail part that is there, it is the light myromycin component. So the heavy myromycin component, that is the head and the short arm, it projects outwards at regular distance and at an angle from each other from the surface of a polymerized myosin filament. So I told you myosin filaments are polymers, right? They are made up of many monomers of myosin proteins. Now here there is a head-like projection. Again here the tail will come. Again here the head will come. Again here the tail. Again here the head. So that is how they form a chain-like structure. Now you might have understood. So they are every now and then there is a projection in a polymer of myosin filament. There is a projection of the HMM, which is called as the heavy myosin monomeric unit. And the globular head is an active ATPase enzyme synthesis site, and therefore because it synthesizes the globular head of the myosin protein, because it synthesizes the enzyme ATPase enzyme, it is the binding site for the ATP, that is adenosine triphosphate, and also it is the active sites for actin. The actin to slide over to, for that also this is the active site, and not just that, for the binding of the ATP also it is an active site because it produces the enzyme adenosine triphosphatase enzyme. So that is the reason why it is the active site for ATP binding. So this was about the two important proteins, and in the previous session we understood why these proteins are very important in the light band and the dark band respectively, because contraction and relaxation are mainly because of these two proteins along with the functioning of the nervous system. So that is why if you have not understood, please go back to the previous sessions and have a look into it. So that is why these two proteins are very important, and we have learned the structure of these proteins. Next, moving further, we shall study about the mechanism of muscle contraction. How exactly the muscle contraction occurs. So here the mechanism of muscle contraction is based on a theory which is called as the sliding filament theory. Of course here sliding occurs, right? We have studied that in the previous session, wherein two actin filaments will slide from covering the myosin filament when in the resting stage. When they are in the contraction, they will slide apart. So all that we had studied. So what is the sliding theory? We shall look into. So sliding filament theory states that contraction of a muscle fiber takes place because of the sliding of the thin filaments over the thick filament. So what happens during contraction? The sliding of the thin filament over the thick filament will take place. What is the thin filament here? Thin filament is the actin filament. Thick filament is the myosin filament. So therefore, the actin filaments will slide over the myosin filaments, resulting in the contraction of the muscle. So that is what this particular sliding filament theory states. So muscle contraction is initiated by a signal that is sent to the central nervous system via a motor neuron. So we had studied about all this in the previous session, right? Simply the muscle contraction cannot occur. Now I want to voluntarily move my hand. So as and when I am moving my hand, within that span of second, signal should be sent to my brain through a motor neuron stating that this person is stretching the hand. Allow the sliding of the actin filament over the myosin filament. That is why I am able to stretch. Otherwise, if proper signals are not sent to the brain, my muscle will not stretch. I will not be able to easily move my hand like this. So that is why they are very important here. And a motor neuron along with the muscle fiber is connected to it, and it constitutes a motor unit. So therefore, the muscle fibers has a lot of connections to the motor neurons. These motor neurons are connected to the central nervous system, and via these motor neurons from the muscle fibers, signals are sent, like how we can talk to one another using a mobile phone, right? Just like that, here the motor neurons acts like a mobile phone, sending messages from the muscle fibers to the central nervous system, stating that this person is moving the hand, promote active sliding of the actin filaments over the myosin filament, so that without any problem, with great ease, the person can move the arm or move the muscle. So that is how it occurs. So the junction between a motor neuron and the sarcolemma of the muscle fiber. So what is the sarcolemma of the muscle fiber? Again, this we had studied in the previous session, right? It is nothing but the covering or the membrane that covers the muscle fibers. We had studied that the muscle bundles are there, which are called as fascicles. In each fascicles are made up of muscle fibers. So that each single muscle fiber is covered by a membrane which is called as sarcolemma. All these we had studied, and it is called as the neuromuscular junction or the motor end plate. So that junction between the sarcolemma and the motor neuron. So what is there? A muscle is there. What is there in the muscle? Muscle fibers are there. What is covering the muscle fiber? The sarcolemma or the membrane of the muscle. The junction between the sarcolemma, that sarcolemma or the muscle fiber is connected to motor neurons. So the junction between the motor neurons and the sarcolemma is called as the neuromuscular junction or the motor end plate, and therefore from the sarcolemma the signal will be sent to the motor neurons. The motor neurons will carry the signal to the central nervous system. The central nervous system will work on it and send the signal back via the motor neuron to our muscles, that is the sarcolemma, stating that this proper sliding of the actin filaments over the myosin filaments has to occur if the contraction of the muscle has to occur. So that is how the movement actually occurs. So this is about the sliding filament theory. So all this is the same thing which we had put the H zone, the I band, the A band. I band is nothing but the actin filaments, A band or the thin filaments, A band is the myosin filaments or it is the thick filaments. So we had studied how the sliding movement actually occurs. So the actin filament slides over the myosin filament. So that is how the contraction occurs. So moving further, now we learnt about the different things. We learnt about the thick filament, we learnt about the thin filament, we learnt about the motor neuron junction, that junction present between the sarcolemma of the muscle fiber and the motor neurons, that is called as the motor neuron junction. All that we have studied. Now how the signal passes we shall look into. So a neural signal that reaches this junction, it will release a neurotransmitter. So don't think that how messages are passed in the form of radiations, like how we speak in the mobile phones, that is through radiations the messages are passed, right? But here how is the message passed from the sarcolemma of the muscle fiber to the motor neuron, and from the motor neuron to the central nervous system, and from the central nervous system back via the motor neuron to the sarcolemma. It is by a chemical which is called as acetylcholine. So acetylcholine is a neurotransmitter. It transmits neural signals for contraction of the muscles, and this acetylcholine actually generates an action potential in the sarcolemma, and this action potential actually spreads through the entire muscle fibers. Is it enough if only one muscle fiber receives the signal? No, right? Or because there are a lot of muscle fibers which are present. So each muscle fiber should receive the signal. So therefore the signal in the form of neurotransmitter, which is a chemical called as acetylcholine, will travel to the other muscle fibers as well, and it will cause the release of calcium ions into the sarcoplasm. Where is the sarcoplasm present? Sarcoplasm is nothing but the cytoplasmic content of the muscle fiber, right? So the sarcoplasm is covered by the sarcolemma. So into the sarcoplasm, the calcium ions will be released, and this increase in the calcium ions leads to the binding of calcium within a subunit of troponin. Now, where is troponin present? If you remember, troponin is present in the actin filament, right? It is a component of the actin, and we had studied troponin is distributed here and there on the tropomyosin protein, right? So, now the increase in the calcium. What caused the increase in the calcium? The signal. The signal in the form of chemicals from the central nervous system was sent to the sarcolemma in the form of acetylcholine, and this created an action potential in the sarcolemma. What kind of action potential? The contraction or the sliding of the actin over the myosin. Now, this signal will spread to the entire muscle fibers, and in the entire muscle fiber at a time, the sliding of the actin over the myosin filament will take place. And this will actually cause the release of the calcium ions into where? Into the sarcoplasm. Now, what is there inside the sarcoplasm? Calcium ions. And as and when the calcium ions get filled in, the calcium level in the sarcoplasm increases. This will actually lead to the binding of the calcium ions onto the protein subunit on the actin filament, which is called as the troponin. And this, therefore, it will mask the active sites for myosin. So, when it masks the active sites of myosin, what happens? The muscle will return back to the relaxing state here. So, utilizing the energy from ATP, and from where is ATP produced? ATP is produced in the globular head of the myosin protein, right? That ATP's energy will be utilized by these actin filaments, and the myosin head now binds to the exposed active sites of the actin to form a cross bridge. So, this is what actually occurs. Now, so much we have understood. Now, after that, when the myosin filament forms a cross bridge, it pulls the attached actin filaments towards the center of the A band. So, if you can see here, it will start pulling the actin filaments towards the center. Now, this is the myosin filament. This part is the myosin filament. Now, you can see the actin filament sliding over the myosin filament. This is the one that is actually causing the contraction of the muscle. So, the Z line. So, what are the Z lines? Z lines are the ones that are present on either side of the actin filament. Again, to understand this, please go back to the previous session and have a look at it. So, the Z line attached to these actins also gets pulled inwards. Therefore, it will shorten the sarcomere, which is called as the contraction. Now, it is clear from all these steps that because of the shortening of the muscle, that is because of the contraction of the I bands, because of the contraction or because of the shortening of the muscles, the I bands will get reduced. That is, the isotropic bands will get reduced, whereas the A band will retain the length. So, that is how contraction of the muscle actually occurs. So, moving further now, what happens in the myosin? Myosin had released ATP. ATP that was produced in the globular head of the myosin was utilized by myosin, right? Now, it is adenosine diphosphate. Why is it a diphosphate? Because already ATP has been utilized. So, from triphosphate, it will get converted into a diphosphate. Now, the myosin releasing the adenine diphosphate and the inorganic phosphate goes back to its relaxed state. And what happens? Again, ATP should be generated. Therefore, a new ATP will bind to the cross bridge, bind to the myosin head, and the cross bridge will be broken. When will the myosin cross bridge be formed? The myosin cross bridge will be formed when the ATP releases energy. Now, ATP has released energy. Now, is there ATP in the myosin globular head? No, right? What is there? Triphosphate became adenine diphosphate. Now, after this process, the muscle will come to a relaxed state. Now, again the stretching has to occur, because stretching, contraction, relaxation is a continuous process here, because we keep using our muscles, right? So, that is the reason. Now, again very soon a new ATP will come and bind to the cross bridge. Therefore, the cross bridge will get broken. and the ATP is again hydrolyzed by the myosin head, and the cycle of cross bridge formation, breaking of the cross bridge, again that will continue. Again what will happen? ATP will be utilized, myosin cross bridge will be formed. When myosin cross bridge will be formed, what happens? The sliding of the actin filament will take place, contraction of muscle will take place, then very soon when the ATP goes and binds, again relaxation will occur, again the ATP will release energy, again contraction will occur. So this is a continuous process that goes on in the muscles, and the process continues till the calcium ions are pumped back to the sarcoplasm, because presence of calcium ions inside the sarcoplasm or inside the sarcolemma is very, very important, because this calcium ion is the one that actually helps in the sliding of the actin filament, and therefore it results in the masking of an active filament. So that is why these are very, very important. So therefore this particular mechanism is a continuous process that occurs in the muscles, and this causes the return of the Z lines back to their original position, which is called as relaxation. So we all knew that the Z lines that is present on either side of the actin filament, they had shortened, right? They had shortened the actin, which led to contraction. Now after all this process, until the calcium ions are available, it will continue. But once the calcium ion depletes, they will come back to the relaxation process. So the reaction time for the fibers can vary in different muscles. That is true. It can vary because some people are idle, they don't do much of the work and their muscle activity is very less. In such cases, the formation of the cross bridge and all that will take different timings. But in especially in athletes, wherein they are active throughout, in such athletes, sports person and all that, a lot of muscle activity is needed. Therefore, here this particular process of muscle contraction relaxation will take place within seconds. It will occur in seconds. So repeated activation of the muscle can lead to the accumulation of lactic acid. So what happens here? At such conditions, because of the, especially in athletes, because of the unavailability of the oxygen, it will lead to anaerobic kind of respiration, wherein the glucose gets converted into lactic acid and the accumulation of lactic acid will take place, and it will result in the anaerobic breakdown of glycogen in them, causing fatigue. So this is one of the important component here. So here therefore, when compared to an athlete with that of a normal person, the functioning of the muscle tend to vary. So this was about the mechanism of muscle contraction. Not just that, we also studied how the relaxation of the muscle occurs, and it is a continuous process that is going on in a body. It is a continuous process, right? So if I'm standing here, I'm talking continuously. We're talking continuously, muscles of my tongue, there also this process tends to continue continuously. So that is why it is a continuous process. So this was about the mechanism of muscle contraction. Next, moving further, we shall study about the types of muscles. So there are actually two types of muscles. One is called as the red fiber, or it is called as the aerobic muscle, and the other one is the white fiber. We shall look into it. First, talking about the red fibers or the aerobic muscles. So muscle contains a red colored oxygen storing pigment called as myoglobin. So myoglobin is a red colored pigment which contains oxygen. If that is present in abundance in a muscle, then that will give the red color to the muscle. So based on the color, they are called as the red muscles or the red fibers or the aerobic muscles. Why aerobic muscles? Because oxygen is present there. So myoglobin content is high in some muscles, which gives it a reddish appearance. And these muscles also contain plenty of mitochondria. Mitochondria, we all know that they are the powerhouse of the cells, right? And these mitochondria utilize large amount of oxygen stored in them for the production of ATP. So that is why they are called as the aerobic muscles. And where is the oxygen stored? Oxygen is stored in a pigment called as myoglobin. That myoglobin is present in the red muscle. In that myoglobin, what is there? Oxygen is there. That oxygen is used by the mitochondria of the red fibers to generate ATPs. These muscles therefore are hence called as the aerobic muscle. So this was about the red fibers or the aerobic muscle. Moving further to one more type of muscle which is called as the white fibers. So some of the muscles, they possess very less quantity of myoglobin and therefore they appear. So less quantity of myoglobin means less amount of oxygen. So therefore they appear pale or whitish in color, and because less oxygen, less ATP production. Therefore, there is less mitochondria in these muscles. So, number of mitochondria are also few in them, but the amount of sarcoplasmic reticulum is very high in them. So, where is the sarcoplasmic reticulum present? It is present in the sarcoplasm, right, of the muscle fiber, and they depend on anaerobic process for energy. That is why it can also be called as the anaerobic muscle. So, this was about the two types of muscles. So, this was about this session wherein we studied about the structure of the two important proteins, that is the actin protein and the myosin protein. So, you might have understood why learning the structure was important. Because we understood the structure of those two proteins, it was easy for us to understand the contraction mechanism of the muscles. And also we learnt about two important types of muscle, that is called as the red fibers and the white fibers. So with this, the session comes to an end. In the coming session, we shall discuss about the skeletal system and the disorders that can occur in the muscle. So we shall meet again in the coming session. Thank you.